Good morning, everybody. Welcome to the Nova package course, which is hosted by the Minerva Center for Movement Ecology and more generally the Hebrew University of Jerusalem. The course will be taught by Wayne and Richard and Nick.
Yeah.
And it's, as you know, a whole week, 5 days. And just to introduce Wayne, Wayne is from UC Berkeley, very famous ecologist, very prolific, wonderful partner and mentor. And he has done lots of work on epidemiology and ecology in general, and behavior and movement ecology. He joined us in the movement ecology year. He was with us at that time for 6 or 7 months in the Institute for Advanced Studies, and now he's keen to share his suggestions and utilities more generally towards computing of biological processes, I would say, very general. And ecology is one of them, obviously, and movement ecology as well. So we thought that it will be good to start with some introduction, and before we do the introduction, everyone will say a few words about.
So maybe we should wait for that introduction when Richard and Nick are here, so I can stop a few minutes and then we'll go around. I think that would be best, so they can meet everybody as well.
Good. So just let me thank Ron. Ron Chen is the coordinator of the course, and everything will work fine because of his efforts, and so thank you, Ron. And I think we can start.
Okay. Am I supposed to switch this on?
It's okay.
It's on, okay. So it's a really great pleasure for me to be here. And I certainly wouldn't be here if Ran hadn't invited me to Israel. I can't even remember, it was 2006. And we had a wonderful movement ecology group at the Institute for Advanced Studies. So before that I was doing a lot of work in disease ecology and some movement because of my disease ecology work. But I worked on African buffalo in the Kruger Park and the spread of bovine TB. And more recently we've been working and mainly finished up on the spread of anthrax in large herbivores in Etosha National Park. And also with Run and All, we worked on vultures. So we've had a wonderful collaboration. Shili has visited my group. I don't know if you know David Zaltz from Ben Gurion. He's visiting me now, so I had to leave him behind. It's very strange. He's there, I'm here. But I should give you a little bit of background. What are we really trying to do here? Why are we doing this? And so I'm going to ask you some questions in order for you to understand better where we're coming from and what we're thinking about. So firstly, how many of you know a programming language like Java or C++? Okay, but there are quite a few who don't, so it's like maybe 40%. How many of you have used MATLAB or R? Something like that, so that's a little bit more. Okay. So how many of you have used dynamical systems packages like Stella or Berkeley Madonna? How many of you have used NetLogo? Okay, so not many. The idea is that today, if you look in population biology, there are a lot of people coming up who are not expert coders. And in order to become an expert coder, you have to work hard, you have to take computer science classes, or you have to work on your own. But you have to really work at it, and you have to use a language to become proficient, and to really develop research models requires that you invest a lot of time in coding. And not everybody is an expert at doing this, and some of you are thinking more about biological processes, and you don't really want to devote half your life to learning how to be a very good coder. So that's why packages like Stella, Berkeley, Madonna, which are graphically driven, and you can program them very easily and very quickly. And I use them in my classes in California to teach people who've never done any coding. And when you build the models, you build them in a graphics environment, and you don't really need to know how to code. If you use something like NetLogo, which is to build, it's focusing on agent-based modeling or individual-based models. So how many of you have built individual-based models? Okay, so by the end of the course, everybody will have done this. So Richard, who's going to come, is he'll be here in 5 or 10 minutes. He looked at programs like Stella, which are very good for simulating dynamical systems. So if you're looking for systems of differential equations or systems of difference equations, and you want to simulate processes described by these equations, and you don't want to code them up using MATLAB or using some more basic language like Python or Java or C++, or even like me, I started out using Fortran in 1970 or whatever. Then you use a platform like that and you can build simulation models. So how many of you have built dynamical systems models? Anybody? Any ecology, prey-predator models, anything? Models, population viability analysis. So who's built some models here? Okay, what sort of things have you done?
Decision making, orientation.
Oh, okay.
And some, well, I didn't build the model, but I adjusted like dispersal.
Okay. So what did you use to code to?
MATLAB.
MATLAB. Okay. Yeah. So MATLAB seems to be popular in Israel for some reason. Maybe you like to pay money. I don't know.
No, that's the reason it's free for Israelis.
What, MATLAB?
Yeah.
Why is it free?
Give it for free for third world countries.
Oh, really? Okay. Well, in America, we have to pay so much money for MATLAB that all the ecologists now use R. And R is wonderful. But you have to also, there's some nice interfaces for R and front ends that you can use that make it easier. But Richard, who saw these programs like Stella and Berkeley Madonna, and also NetLogo, which is agent-based, so that's a different paradigm, he thought, why isn't there one framework that does this very easily? And because I'm interested in these things, Richard visited me and we decided what we're going to do is we're going to try and develop this Nova platform that he had started working on so that it can serve the interests of people who are working as scientists in population processes. But of course it's also the social sciences, anything, and even engineering. But engineers usually are much better at coding and they like to use MATLAB. And MATLAB's very good for them, and they also program in basic languages. So we were really developing this Nova platform to take the pain out of coding, so that scientists who are really regarding themselves as population biologists, but who really do need to do more computational work now, because there's a lot more data. If you look as movement ecologists, which I assume most of the people here are in movement ecology and have interest, is that correct? Or is anybody pretending to be a movement ecologist who's secretly something else? I mean, it's okay. You should admit to it now, because then we can help you work in your particular interest. So we collect a lot of data using GPS, and we try to interpret this data, and we try to say, what does this data mean? And one of the tools we have for helping us understand what the data may mean is to simulate data using different processes and saying, how does the simulated data compare to this data we're getting? And are there patterns emerging from our simulated data that look like the patterns in the data that we're analyzing, and so can we make some potential inferences about the possible processes that are creating our data. So is Richard coming? Okay. So we're going to go around and introduce everybody once Richard comes.
That sounds great.
So the idea was to develop this Nova platform. Okay, so here's Richard. And I'm just doing an introduction now, and then we're going to go around and everybody's going to introduce themselves. So I'm just explaining a little bit about the background and why we're here and why we're doing what we're doing. And so we're interested in facilitating research and teaching through a single platform. We wanted to develop a platform that we could use for teaching, so it has to be very simple. We want to be able to take people who've had no coding experience at all and start them building models. And this I do in my undergraduate class at Berkeley. But once they get into this platform, we don't want for them to hit a ceiling and say, okay, now I have to go to some other language, something more sophisticated, some full language like C++ or even MATLAB or R, where you can do almost everything you need to do. Because if you start off using and learning Nova, you want to stay with that. But there are a couple of important things about Nova, important goals that we have. So one of them is we want to make coding very efficient and very accurate. And we want people to be able to collaborate with one another and to be able to share code and build on what others have done. And so we want to develop a community of users so that we can all benefit, so that when you want to simulate some data, almost everything is there for you. And if there's a community of users, everything will be there for you. So as you'll come to understand as we go along, Nova is like, I don't know what you call, yeah, when I was in South Africa, we had these things called Meccano sets, or there's constructor sets. Erector sets. You know what I'm talking about, where you have pieces, but also breadboards with little electronic devices that do things. So you make your electronic whatever by putting different components together. The idea with Nova is that eventually you won't have to develop any code. We have these things called code chips, and people have modules. And you want to be able to reuse elements that perform certain tasks, and you want to be able to hook things together to make more complex models. So as we go through this week, you'll see how this is all done in Nova. Now what you have to appreciate is that we're at the beginning of developing Nova. We're at the alpha level, we're not even at the beta level yet, huh?
I'd say the beta.
We are? Okay.
We wouldn't be able to do this with him if we were at the alpha level.
Okay, we're at the beta level, but some of you might feel a little frustrated from time to time because things don't work exactly right. But we're certainly moving forward as rapidly as we can to turn this into a program that, as I said, takes the pain out of coding for you. That if you want to do something, you'll be able to do it much faster, much quicker, and much more accurately in Nova than any other platform. And the important thing about accuracy is that if you're reusing bits of code that are tested, so it's like if you go to R and you do a statistical test and you import a procedure or you do something, many people have used it before and it's been tested and it's got a certain level of reliability. And so you don't have to reinvent the wheel. But you do have to put some code together, and whenever you put your own code together, you have to check it very carefully. Have I made some stupid mistake? Is this garbage? And then you have to check the output. Does this make sense? And you have to go through this whole process. So if you have a platform where you're using elements that are already been tested to do certain things that you know work, and you can just hook these things up together, then you can very rapidly build powerful code. And you don't actually have to do any coding. So that's the ultimate goal. So what Richard, Nick, and I decided to do is that we wanted to show people what we have, and the best way to do that is in workshops. We're interested in building different kinds of models that people can use in movement ecology. We're also doing the same thing in disease ecology and general ecology. And if you went to the Nova website and you looked at novamodeler.com, you would have seen that we have various models. These are the usual toy models that you'll find if you go to NetLogo, if you go to others that people use in the classroom to demonstrate certain concepts. Things like, at the most elementary level, the game of life. Has everybody here seen the game of life? Have anybody here played it on a hexagonal array? So we're going to do that. We're going to play the game of life on an hexagonal array, because we have both rectangular arrays and hexagonal arrays. We move points over the plane. We do a lot of different things. So I think that's enough of a general sort of background of what we're trying to achieve here. Did we want to maybe go around and people can say where they're from and what they hope to get out of the course?
Yes, please.
Yeah, okay, so let's start there.
I'm Idan. I'm studying currently in Sde Boker under the supervision of David Zaltz, who left me for you.
He's going to come back.
And I'm here to deepen my knowledge of movement computing.
I'm Rand. I'm heading the movement ecology Berkeley in the university. I'm the director of the Minerva Center. And unfortunately, I will not be with you most of the time, but I trust you in good hands. And yeah, that's it.
Me? Oh, I'm Richard Salter. I'm on the faculty at Oberlin College in Ohio, and I guess I'm responsible for Nova. So if you don't like it, you can yell at me. If you do like it, you can yell at him.
You can praise me. Yes.
I'm a PhD student at Rand's lab, working on jackdaws and connections between social status, social rank, movement, fitness, and plant dispersal. And expecting to get also a tool and some theoretical basis in this workshop.
My name is Sippl-Swezey. I'm a research specialist at the University of California, San Francisco. When we mentioned doing work in disease ecology, he was mentioning some of the work that I've done in looking at human disease transmission and clinical trial interventions to reduce transmission of infectious disease. That's my domain at UC San Francisco, along with general modeling. I've used Nova for a good number of years now, and I think it's a very powerful platform for rapidly building detailed, specific, complex models that are intelligible and communicable. I think this is going to be a great week, and I'm looking forward to working with all of you.
I'm David. I'm doing a PhD in Rand's lab on the movement ecology of food bats and how sociality and social factors affect foraging movement decisions in the food bats.
I'm Shai. I'm also a PhD in Rand's lab, and I'm working on white storks. I've contacted you by emails, and I need to thank you later for the help. And I'm working on movement ecology and behavioral ecology of white storks, trying to connect movement to fitness, and I hope I can get some basic ideas of how models can help me understand my empirical data.
I'm Minerva. I'm in a master's Genetics lab in Eilat. I've just started my master's, and I'm interested in behavioral ecology and decision making in school of fishes, and I hope to learn stuff that can help me later.
I'm Nana. I'm a master's student in Rand's lab, and I'm working on stone turtles, on navigation and learning. And from the tutorials, I really like the Nova feeling. So I love to meet you.
Thank you.
I'm Shirley Balbrid. I'm a researcher at BGU, Berkeley University in Berkeley campus. I used to be a postdoc at Getz lab. And I'm working also on movement ecology, and mainly on the wild earths in the Negev. They were reintroduced there, using also genetic tools. So I hope to get some overview on the program.
I'm Tarin. I'm a PhD student at the Faculty of Agriculture in Oberlin. And my PhD is on omnivorous lady beetles and their functional behavior and fitness from different food types. And I'm hoping to use NOVA to try and create a model that simulates movement between plant-based foods and prey-based foods.
My name is Ecology. I'm from our non-Times lab in Tel Aviv University, and I work mostly on learning processes and the evolution of learning mechanisms. And I just like to learn whatever you have to offer.
I'm Guy. I'm also from Tel Aviv University, from Amir Ayali's lab, and I just started my master's degree. It will be on locust motion, collective motion.
I'm Ro'i. I'm a PhD student in Ran's lab, and I'm working on griffin vultures, social behavior, biomechanics of fly, and I hope to study a tool, and also, as was mentioned here, some more about modern theoretical work.
I'm Yotam. I'm a PhD student in Ran's lab, and I'm working on stone curling navigation. And yeah, I hope to see what can we do with this tool.
I'm Nir, also at Ran's lab. I'm working on invasive plants in China, and also hoping to learn some modeling tools.
I'm Eigal. I'm a student under the supervision of Moshe Kiflawi in Eilat, also Ben Gurion University. I'm working on the early stages of coral reef fish, how do they find their way, and I implement current maps, etc. And I'm interested in modeling, so I'm happy to be here.
I'm Noam Joseph. I'm a PhD student in Nadav Shashar's lab. I'm modeling camouflage patterns and trying to quantify camouflage as a reaction. So I'm not in the field of trajectory and motion, but I'm willing to get my hands dirty in that field. So I want to learn and to see if I can combine the two fields and to see how it affects one another.
I'm Gabriela Escata. I'm as well in Nadav Shashar lab, and I'm doing a master in marine biology and with a topic on spatial learning and navigation in cephalopods. So I hope I can learn something useful.
I'm Ranel in Yoni Bellmaker's lab in Tel Aviv, using acoustical telemetry to follow movement and grazing patterns of invasive rabbitfish. We mostly use R. I found it very difficult so far, so I'm hoping I can find a way out by...
We also hope so. I'd like to give you a guarantee, and I will almost give you a guarantee. But we'll have to see.
I'm Adi, study in Haifa. I work with sharks. I usually, but until now I did mostly genetics and general ecology, but now I want to get into more movement ecology, so I'm studying more development.
Is that everybody? Okay. Okay, so the general format is that I will give every morning a lecture of variable length, hopefully not too long, but just giving the background of what we're going to do. And I think you've all seen what we're trying to cover. So like today, what we're going to talk about is the basic statistical data that you get from recording where animals are on the landscape, and you can calculate two kinds of distributions, a step size distribution and a direction of heading distribution. But that's just pure information from the trajectory, there's nothing else. And so we talk about this in the context of different kinds of stochastic processes, usually referred to as random walks. Then tomorrow we'll get into things that have to do with the fact that animals are not always doing the same sorts of things, so there are different phases of the trajectory where sometimes the movement reflects heading in a direction, maybe towards water or maybe towards going back home or something like that. In another, it might be a foraging behavior. And so you get mixed distributions in creating your data pattern, and we're going to talk about that. Then we're going to, and that's following individuals and how they move around on the landscape. And NOVA has the ability to obviously just map individuals on a landscape in a continuous way, but it's not entirely continuous because obviously whenever you do something numerical, you have to discretize it to a certain level of resolution. Then on day 3, we'll start to get into agent-based systems where individuals are moving around on landscapes. And again, there are 2 ways of doing this. You can take an individual and you can move them around on the Euclidean plane just with an x, y coordinate, or if it's a vulture, x, y, and z. So we're not going to do anything in the 3rd dimension in this particular workshop. But on the 4th day, we're going to start to look at how agents make decisions on where to go. And we're going to have different rules. And then we're going to show how NOVA can be used to evaluate the utility of different rules in terms of how efficient the animals are at collecting resources if they follow those rules. And we'll do them in the context of genetic algorithms. So I'll introduce genetic algorithms on the 4th day. Ron was very keen that we got into this. Has anybody worked with genetic algorithms? And then on the 5th day, we're going to talk about more sophisticated ways for making decisions using artificial neural nets. Now, your grade for this particular course is going to be your final project. And for your final project, we want you to at least have a genetic algorithm working and evaluating what the best strategy is for an animal to make decisions on moving on a landscape using genetic algorithms. And you can either do this with a simple rule, or some of you may get a little further and want to actually use the neural nets. But it's not going to be necessary for you to do the neural net stuff as part of your final project. Are there any questions? Do you want to make any comments about the final project or anything like that? Okay.
Maybe we should say that the course will not be here, right? Also not the morning lecture. All the course will be in the computer center.
Yes, it will be, and we have to start at 8:45. Is this
8:45 in the computer center tomorrow. Okay. Yes. So as I said, I'll give it a variable length, hopefully not too long, and then either Richard or Nick today will talk about how to use NOVA to put together the kinds of models we want you to use to do your exercises in the computer lab. Then in the computer lab, you will build your NOVA models and you'll generate data. Now, one of the things that NOVA can do, which we won't be doing, and that is NOVA can be interfaced with R, and you can prepare data for NOVA using R, and you can ship data that you've generated through a NOVA simulation to R. Some of you who are very good with R, maybe towards the end of the week, Richard or Nick may show you how to put those together. But in the exercises today, the data you generate in NOVA, you can save as a CSV file. And then, I don't know how you analyze your data, whether you use stat packages, who uses Excel. What do people usually use to analyze their data? MATLAB. So then you can take this data and you can ship it and you can bring it into MATLAB to analyze it, build histograms, do your plots and things. And then you might say to me, well, why didn't I do the whole thing in MATLAB? And the reason why you didn't do the whole thing in MATLAB is because when we finally have a community of users using NOVA, you will be able to come to a problem and say, I want to do this, and within a couple of days you'll have it done, whereas if you're using MATLAB, maybe some months later, you'll have it done. So the idea is that Nova is going to enable you to get to where you want to be in generating your simulations an order of magnitude faster than anything else. And that's going to be because you have reusable code chips, reusable modules, and these things have been tested, and the computer generates the code for you. You don't have to write any code unless there's a little code chip that you need to do something that's never been done before. And then you write it in NovaScript, which is really an extension of JavaScript. So, and for me, if I sometimes write these little code chips, I don't know any JavaScript. I just look up on the web, what's the right syntax for, you know, conditional statements in JavaScript, and you can do it. I mean, it's at that level, that's the amount of coding you need to use. Okay, any questions about anything before we move on? Okay. So I've called each day a module, and we're going to have some theory objectives for today. Should I turn the lights down there? Okay. So the first idea is an animal moves continuously through space, and we have devices to tell us where these animals are, and they're like strobe lights. Every so often the light flashes and you get a position, whether it's a GPS collar collecting information. I have a colleague in Germany who's using radar to track little bees flying around and can do this with a radius of 2 miles. There are other methods for collecting data, I guess, new technologies coming along. But basically we're talking about an animal moving through space, and we're just collecting these pictures at points in time at regular intervals at a particular frequency. Where's the animal now? Where's it next? Where's it next? And so from these, if you know where it is now and where it's going to be next, you can calculate two things. You can calculate the distance between the 2 points and the direction in some coordinate system. So you get these so-called step size distributions, and you also get turning angle distributions. Now, if you go through the literature and the step size distributions, you'll see that people talk about the uniform, a power distribution. I talk about a beta maybe more than most people. Normal, log normal, also Weibull. And why Weibull? Why? It turns out that there's a good reason, and I'll come to that in a minute. And then there's so-called truncated distributions. So truncated distributions are distributions that have infinite tails, either on one side like a log normal, or on both sides like a normal. But we know that the step size distribution probably has a maximum. The animal cannot go faster than this. So why would we have a tail to infinity? So we can cut it off. So people sometimes take that into account. Then turning angles. So the thing about movement is because we have a strobe light, and our first instinct is to just join the 2 points by a straight line. So, you know, you go there and then the next point is over there. You can look at the angle that's been made. The animal is not going on the same path anymore. It's deviated somewhat. So you can measure that deviation, and you can get turning angle distributions. And in the literature, again, there's uniform distributions. There's a thing called a von Mises distribution or Rapp-Cauchy distribution. Is anybody here familiar with the Rapp-Cauchy distribution? Okay, just a couple of you. Who's seen a Weibull distribution? Okay, some more. So what I'm going to do is, I've got these distributions up here. We'll go through them very quickly. The idea is not to teach these distributions, but just to have them in this lecture for you if you want them. This is now online. So if you go has everybody been to the Dropbox? Is anybody here having problems accessing the Dropbox? So at the Dropbox, I've got 13 or 14 papers. I've now put this first lecture there, so it's there for your reference. Then we'll talk about how people take from this kind of data, the strobe lighting data that they have in these distributions, they generate histograms or different kinds of plots, and they try to interpret what's going on from these plots. But one of the things that I want to stress in our lecture today, and in the exercise that I give you, that very often the data that you get depends on the frequency with which you sort of flash your strobe light or you examine the trajectory, compared to the underlying processes that are creating that trajectory. So there may be very fast processes or somewhat slower processes. So by a relatively slow process, it depends on your time scales, but for example, on the African systems that we work on, the zebra may be grazing in one area and then head to water maybe 5 kilometers away. And so for the next hour, they're going to be walking in a particular direction. So they have walking in a very directed way. And then one of them, if they get chased by a lion, that may be over in a few minutes, and they may be dead, or they may escape. And so there may be a dash and an escape. And if you are sampling this pathway every 20 minutes, then there's certain things you may miss. If you're sampling it every 10 seconds, then you'll see a lot of different things. So we're going to try to understand how the relationship between the frequency of collecting things and how you bin your information. So, if you collect information on step sizes every so often, and you have a range, but you don't have so much data, you just have 200 or 300 points, and you want to make a histogram, and some step sizes may be a few meters, and some may be a few hundred meters, then how many categories should you have to get the frequency of steps between like 5 and 10 meters, 10 and 20 meters? Should you plot that on a log scale? What should you do? And how will those choices affect what the distribution and what the data look like to you? Because they all have an effect. So we'll talk a little bit about that. And then the NOVA objectives will be to introduce you to NOVA as a graphic tool, using distributions to simulate strobe data. So we'll say, okay, we're going to draw step sizes from a uniform distribution that's uniform on a particular interval, and we're going to create data. And then we're going to analyze that data. And as I said, this process is very useful for understanding how the way you analyze the data relates to your understanding of what are the processes that create the data. When you create the data, you know what the process is. So when you do something to it in analyzing the data, you can understand how your analysis either gives you a good view of the underlying process or distorts that view of the underlying process. And when you make more sophisticated models and you add more processes, you say, when I analyze the data, those processes are going to create patterns. What will those patterns look like in my analysis? How do they compare to patterns of empirical data? And can I reject certain processes as being inherent in the empirical data, because when I put them in my simulated data, they create patterns that I don't see in the empirical data. And then we'll explore the effects of subsampling your data to get inferred distributions of what really caused the data. And that'll become clear as we go through this lecture. Okay, so you see there's the green path at the top, and so let's say this is the actual pathway, so it's not connected by straight lines, the animal is moving around, and then your strobe flashes at a regular basis, so obviously from here to here the animal hasn't gone very far, so it's moving very slowly there, here it's moving faster. So here you can see a pathway where I'm now looking at the pathway every t units of time. If I were to look at the pathway not every t equal to 1, but every second observation, and if I were to get step size distributions and turning angle distributions, then I would generate a data set. I could also look at what's called the distance of the individual from the starting point. So you can see that I've got a distance metric that tells me how far I am from the starting point. So there's certain aggregated measures that tell me something about the trajectory as it proceeds, in addition to the two distributions of step size and turning angle. And you see here that if I collect the data every fourth point instead of every point, I'm going to get bigger steps in general. So here's a big step, there's no step size that size. So obviously the biggest possible step depends on how frequently I look at the data. And so you can see already that your choice of sampling interval is going to affect this distribution. So there's a lot of papers in the literature where they have different examples and people have looked at distributions, and you can read these papers. This course is not about us going through these papers because we want to teach you the hands-on stuff, but this is background material for you to read as we go through. So I've selected some of the important papers. And Morales et al. In 2004 published a paper in Ecology where they were looking at data from elk, and they had a number of elk, and this is data from elk 287, and they actually recognized two different phases of the animal's behavior. One which they called the animals encamped in a particular area, and another is the animals exploring its on a much wider horizon, and these behaviors take place at different times of the year. And so here's the daily movement rate in kilometers. So this is like a step size distribution, and you see most of the steps are actually quite small. So here's 1 kilometer, 2 kilometer. So the animals actually stay very local. But when they go into the exploratory phase, then we've got a much bigger scale, 0 to 20 kilometers. And so they've binned the data over here, and you can see that the animals move in one day. A lot of the highest frequency here is to go 5 to 7.5 miles, I think this kilometers the scale is. And so you can plot this data, and you can fit a distribution to these data, and you can also do the same for angle of turning. So if you look at consecutive steps, you can have a look at the change in direction, and you can plot these angles, and you can get different characteristic distributions for different phases of the animal's life. And so if you want to know more about it, you can read this paper. So very quickly, just so everybody is reminded, we're going to go through the different distributions, and we're going to mainly work with the uniform distribution. So the uniform distribution is a function that is defined on an interval AB. It's 0. A can be negative, but obviously if it's a step size distribution, it can't, so it depends what we're plotting. But if we're plotting a step size distribution, A can typically either be 0 or there may be a minimum step size. And then it's a constant equal to a frequency of 1 over B minus A over this interval, so that the area here is 1. And then it goes back to 0. So that's the uniform distribution. And if you have a look at most computer programs, they have this function called random. And random usually draws a number to a particular precision between 0 and 1. And you can rescale that number so that it goes between A and B. So is everybody experienced with that? That's fairly simple stuff. But we're going to be using it in our models. But then drawing values from known probability distributions. So the uniform is very easy. Now, most of you, I'm sure all of you are familiar with the definition of a probability distribution. So here's a probability density function and a cumulative density function, which is the integral of the probability density function. We say minus infinity to x, but sometimes from 0 to minus infinity, from minus infinity to 0, the function may be defined as 0, in which case you can take this integral from 0 to x with x positive. Now, the important thing about a probability distribution, a cumulative probability distribution, is that on the horizontal axis you have the value of x. On the vertical axis you have the cumulative distribution going from 0 to 1. So at some point you've gone halfway. You're at the 50th percentile of the distribution. Or you've now got all x. Is there somewhere I can draw? Is there a board maybe?
Just opposite of that.
Well, I'm sure, can you all imagine this?
Yes.
Yeah, so I've got a cumulative distribution like this, and I'm going along here with my x value, and I look at my x value and I read off what proportion of the graph of the distribution have I covered for a particular value of x. So xp means that I've now covered a proportion p, and I've got 1 minus p proportion to go. So on the vertical axis, what I have is as I move up and I go back to the x-axis, so this is the inverse function, going from p back to x, I have what is called the quantile function. And this quantile function means that if I draw a number between 0 and 1 at random, it gives me the corresponding x for that distribution, such that p% of the points would be below that x, and 1 minus p% would be above that x. So these quantile functions are important in order to be able to generate values of x when you're drawing for your simulations. Now, it turns out that the quantile function requires you to know the inverse of the CDF, the inverse function. This inverse function may have an expression in some cases. So the Weibull distribution, there's an easy way to express this. That's why the Weibull is good, because numerically you can calculate this very efficiently. For others, like the normal distribution or log normal, there are ways to calculate it, and if you look in different packages like R or MATLAB, there may be methodologies to do this. There are people who actually write their own code in C or Pascal or anything, can buy a book like Numerical Recipes and you can do it. And but we're also going to talk about the exponential function and the Pareto function, and those have simple ways to calculate this. So it can be a bit of a challenge depending on the distribution you have, but there definitely are algorithms for doing this. And in Nova at this time, the best way to do some of these is to actually rather do it in R and import the values. But in others, it's very simple. And if you're drawing from a uniform distribution, then you don't have a problem at all. So you can read up a little more about the Weibull function and numerical methods for doing it. As I said, I've got this here as a reference for you. We're not going to use this in any of the exercises that we're using because we're sticking to very simple distributions where you can draw the random variables very easily. But if you want to work with more sophisticated distributions like the log normal, then you need a numerical method to calculate, to generate these values for you. So typically what happens is you draw a number from a uniform distribution between 0 and 1, and you get 0.6, for example, and then you say 0.6 corresponds to what value of x for my given distribution that I'm interested in. And you have to compute that. And as I said, if it's the uniform distribution, you don't have to compute it because you're drawing from a uniform to begin with. If it's a normal distribution, there are tables that you have that can help you compute this. So they're lookup tables. And you can get lookup tables and you can calculate the value for any distribution. And people have been doing this now for a long time, so all that information is out there. Now, one of the distributions that I think is a relatively important distribution is the beta distribution. And the reason for this is that the beta distribution is a compact distribution. It has a minimum and a maximum. And in its simplest form, it goes between 0 and 1. So here's the beta distribution that goes between 0 and 1. And for different values of the parameter A and B, you can get the distribution to look almost anything you'd like it to look like, as long as it's unimodal or it's U-shaped. So for different values of A and B, you can see that you can get something that has one mode and is fairly regular, and you can move this back and forth depending on how you choose A relative to B. You can also give it tails, or it can come down without a tail. So it comes off here with a tail and comes down without a tail. It can start without a tail and come down with a tail. So you can just, for different values of A and B, you can actually do a lot. Yes?
That's a typo there, right? It should be dt.
Instead of dx.
Yeah.
Right. Yeah, so that should be dt. The x comes from the fact that you're going from 0 to x over there, but it should be dt.
You mentioned that you like the beta. Do you like that for step size, or do you like that for theta?
No, yeah, so this is for step size. So here's the Weibull. The Weibull as well has a lot of flexibility. You have 3 parameters. The first this parameter, called the location parameter, in these it's set equal to 0, but what it allows you to do is to shift the distribution so that you have a minimum at x equal to theta. And you define it to be 0 for x less than theta, so you can shift it. But then once you've shifted it, it's got 2 parameters, k and lambda, and for different values of k and lambda, you can make this thing either have an infinite height over there. You have tails going off to plus infinity over here. But depending on how you choose it, the tails might be very, very, very small. And also the Weibull distribution can be characterized in terms of a uniform distribution. And if x is uniformly distributed on 0, 1, then the random variable computed in that way is Weibull distributed with parameters k and x. So this is why the Weibull distribution is favored in movement ecology and simulations, is because you choose your uniform variable by calling your random function, and then you compute this for given values of lambda and k. What value of x did you draw from the uniform? You calculate this thing, and you've got your Weibull distribution simulated. So that's why people like the Weibull, because it's very easy to generate in simulations. Now, one of the things that is very important in movement ecology has to do with fat-tailed distributions. And a fat-tailed distribution is defined in terms of the behavior of the tail. As x gets large, how does the tail behave? Does it behave like 1 over x, or 1 over x to some power? And what is that power? Does it behave like 1 over x squared, 1 over x cubed, so on? So such distributions have infinite mean if alpha, the tail, lies between 0 and 2. So if it falls off like either 1 over x, or 1 over x to the 2/3, or 1 over x to the 1.4, or whatever, then it means you have a lot of a lot of frequency in your tail as it goes out. So when you calculate the mean turns out to be infinite. If alpha is somewhere between 2 and 3, then the mean is finite, but the variance is infinite. So that means a lot of points actually turn out to be rather large. And for the last 2 decades, or decade and a half, I should say, people have been interested in fat-tailed distributions because of the work of a statistician, Levy, who first has a general class of Levy distributions that are not exactly these power distributions. The Pareto distributions are these power distributions that are defined in this way. Levy distributions are defined a little bit differently, but the tails of the Levy distributions behave like this for different values of the Levy parameters. And the question is, do animals exhibit step size distributions that are fitted by a fat-tailed distribution? And if they are, then the patterns of the movement do some interesting things, and we'll talk about that. So it turns out that if you use the Pareto distribution, which is a very simple distribution, that's the cumulative density function for the Pareto distribution, then the quantile function is very easy to calculate. You can do it analytically. So it's very simple to simulate Pareto distributions. But a lot of distributions don't look like a Pareto distribution for small step sizes. They only look like them for large step sizes. So they're not necessarily good distributions to always fit to your data. The Levy distributions come from a family of distributions where this is the probability density function and this is the cumulative density function. It's defined in terms of the so-called error function, ERF. I'm not going to go into the math of this other than to show you that for different values of C, different Levy distributions, this is what they look like. And depending on the value of C, you'll get a fat tail. And you can see some of these tails are going out and they're not coming down to 0 very rapidly. So the reason for the interest in Levy distributions versus fat-tailed distributions comes down to Brownian motion. So is anybody here who knows how the ideas around Brownian motion first entered science? Anybody? Huh?
Einstein.
No, before Einstein, in observations. So people were looking under the microscope. Huh? Brown, yeah. He was looking under the microscope and he noticed that little biological entities were moving around and they thought they had a life force in them. But then they found out that even if you put little dust particles in a liquid medium and you look at them, they jitter. And what could this jitter be? And Einstein was the one who showed convincingly in a 1905 paper, and he called it Brownian motion, that if a particle is small enough, the stochasticity of the molecules of the liquid hitting the particle, sometimes they hit in one direction by more particles than in the other direction. So they're sufficiently small that there's going to be enough variation in the difference between particles coming from different directions that they start to jitter. It's got nothing to do with some mystical life force, but in fact it can all be explained in terms of the particles in, if a liquid is made up of tiny atoms, then this is evidence for the atomic theory actually of a liquid. Now Levy showed that, sorry, let's just go back to the Brownian motion. So Brownian motion, if you look at step size distributions, this distribution does not have a fat tail, it's Gaussian. And a Gaussian function has a finite mean and finite variance. So when you plot the step size, it doesn't have this fat tail. There aren't any extraordinarily long steps that happen with a relatively high frequency. These only happen exceptionally rarely, so that most of the time you look like a string of wool. Sometimes you might get a persistent direction just by chance, or there may be a slightly bigger step size. And so this is what a Brownian motion path looks like. Now, if you have a fat tail distribution, then every so often, with a much higher probability than in a Gaussian model, you take a big step. So here you take a big step, you mess around a bit, you take a big step, you mess around a bit. Now the idea is that if you were sampling infinitely often, then you could get as small a step as you like. And so it is possible, if you have a look here, that this is made up of even smaller steps. If you were to take this region over here and blow it up, it would look like this picture. There would be things that look much longer than the average step size of the little steps in there. There's something that's quite a bit longer than the average step size. Although on this scale it's not long, on this scale, when you blow this up, you get something that looks like this path again. So that's called self-similarity that occurs irrespective of the scale. So with Levy distributions, you get the self-similarity property. And that's interesting for movement ecologists, because movement ecologists are looking at trajectories and they say, gee, are animals moving around like this, or are they moving around like this? And if they're moving around like this, why are they moving around like this? And there's some hypotheses about search. And if there's a certain kind of resource distribution on the space and animals are searching for the resources, and depending on the density, it's calculation to be done that it's optimal to do a levee walk to search for these resources. So when there aren't any resources along, you keep going until you encounter some resources, then you pick them up locally, and then you go off in another direction once you've finished foraging for those resources. Yeah, Nick?
Out of curiosity, what did you use to generate these plots, to generate this data?
I probably grabbed it from Wikipedia. So a lot of the stuff here I've grabbed from Wikipedia.
Yeah, they're not the same scale, that's what you mean. Yeah, this one is much larger than this.
Well, in the Nova workshop, we'll be generating this kind of data. So I was curious how you generated it for this slide.
Stole it.
Yeah, so I could have generated it, and we will generate it. But when I was making my lecture, I don't know, I could have grabbed it from a paper, I could have grabbed it from the web. Sometimes, I usually do it, if I grab it from somebody's paper, I usually do put the attribute where it's come from. So, so here we come to the issue of truncated distribution. So in a truncated distribution, this is the normal distribution. This is the distribution you're using. This is the truncated distribution, which you define to be this distribution on the interval AB. And if you divide by the cumulative value FA minus FB, then this is normalized to be equal to 1. So you define this distribution, truncated distribution, to be the original distribution rescaled on the interval AB, and then to be 0 outside the interval AB. So it's just, sometimes it looks normal, but it's truncated. It's useful to use these in some cases. And there you see the expression below for the truncated normal distribution. So I thought I would just put it in there so we have it. Now we come to turning angles. So if the turning angle is totally random, then you would have a uniform distribution on minus pi and pi. That means the next angle could be anywhere between minus pi and pi. And it actually turns out that the von Mises distribution, which is defined by e to the k cos x minus mu, and then it's normalized And the normalization gives you a function that's called a Bessel function of order 0. Don't worry about that. It's just the integration of the cos functions are called Bessel functions. So it's got a fancy name, but I could have written it in terms of the integral. The thing about the von Mises function is, firstly, where mu is kind of where the distribution is heaviest. So here mu is 0, and this is just plotted for different values of kappa. You'll notice that when kappa equals 0, we get the uniform distribution. And then as kappa is increased from 0, so it becomes more and more peaked around mu. So that's one way of characterizing a turning angle distribution. And so, for example, if an animal basically is moving around, but is preferentially, as it moves around, thinking, I want to get closer and closer to home. And home is in that direction. But I'm going to move around, but I'm going to bias my movement so that I kind of head towards home. Then a distribution for turning angles might look something like that.
Out of curiosity, the plot that you were showing before, the step size distribution plot?
Yeah.
That's with a random turning angle, that Brownian motion and the Levy walk?
Yeah, so the Brownian motion and the Levy walk there, we were talking about step size distributions with random turning angles. We're going to come and we're going to talk about different kinds of stochastic processes or random walks as soon as we finish talking about these distributions. So there's another distribution that's called the Cauchy distribution, and it's called the wrapped Cauchy distribution because you identify this end with this end because it's a circular statistic. And again, it has parameters. This you can set this parameter so that the peak occurs at a particular angle, and then it's called wrapped because the wrap it's not symmetric with respect to minus pi, but it's symmetric if it's shifted by the amount of that angle. And it's also expressed in terms of hyperbolic sine and hyperbolic cosine. So this is a distribution. It's just these, if you read the literature, people will talk about these distributions in movement ecology. Now we come to talking about data, and here's some data from a very famous journal called Movement Ecology. I guess you've all heard about that. Wonderful journal. They publish my papers. Lots of journals I send papers to, and they say, nice work, but too focused, not general enough. So I don't know how many of you are going to become academics, but if you are, you better not be sensitive to criticism, and you better be willing to be called an idiot, because by the time you get your paper back from reviewers, that's what you feel like. You feel like an idiot, the way they say, should have done this, should have done that, you did this wrong, you did that wrong, or at the very best, it's too uninteresting to publish in our journal. But fortunately, the stuff that I do is interesting enough for movement ecology, so I'm very happy.
Rain is on the editorial board.
but that doesn't help. That doesn't help me get a paper in, for sure.
No.
Never. In fact, it makes it harder. Because we have to be seen to uphold standards and not... Anyway, so if you look at a trajectory, you start at the first point, you go to the second point, you go to the third point. So you're beginning to accumulate numbers of points. So here you see a number of points laid down on a trajectory over space. Here the grid size is such that the trajectory falls mainly within a few grid cells. Here the grid size is such that the trajectory falls in a broader range of cells. So let's say you start off with the first point and the second point and the third point. Over here, what you can plot is n of t is the number of points in the trajectory so far. So you start with the first point, and then you ask, how many of the grid's cells are empty. So there are different ways of generating statistics that tell you what's happening with the trajectory. Here's one way of doing it. I'm not saying it's a good way. It's a way that Gautestad, who's an excellent theoretician, and Musterud, who's actually got a lot of experience in movement ecology, are doing it. So if they're doing it this way, then the chances are that it's a useful thing to do. But for the moment, you might say, why are we doing it like this? But so what you're doing is you're generating this curve, and you're saying, okay, how many, I'm going to plot this on a log. So I'm going to take the log of the number of points so far along the trajectory, and a log of the number of cells that points lie in. So if points lie in the same cell, then the log of that is 0, and then you start to get it starts to fill the space out. So you get a curve here which tells you how the space is being filled out as the number of points increase. And so you can plot two different trajectories, and you can say one is filling the space out faster than the other. So this is one way of looking at the data. And if you want to see why did they do it in this way, then you must go to this paper. I would actually not recommend that you start on this paper because it's one of the most difficult papers I've ever read. So now you can plot other things. For example, you can take your step size distribution and your frequency, which is a histogram, but instead of plotting it as a histogram, you plot it as a continuous curve. So you're plotting here the log of frequency and the log of step size. And if you have Brownian motion, then this curve will look something like this. And it's a curve where the slope's negative, getting steeper and steeper. So it may start off positive, but the slope, in other words, the tail, this is now we're getting into the tail region where we're getting to longer step sizes. The tail is falling off very rapidly, so that's why this curve is falling off very rapidly. Yeah, the tail persists as you go out, so that's why it's a straight line. And this straight line over here tells you something about if it's falling off like 1 over x to the alpha, what is that alpha? Then alpha corresponds to the slope. Does everybody understand that? Any questions about that? Okay.
Question?
Yeah.
Maybe it's a little bit basic question.
Yeah. No, basic's fine. I mean, we're here to really introduce people to the topic.
So you look on your animal and you look on your empirical data,
yeah.
You look on this theoretical data of the distributions, and if you see they are looking a little bit like this distribution or this distribution, what is the next step? What you can say from this about your animal?
Okay, so that's a very good question, and I'm going to show you in the next slide some simulation data and what people did and how they analyzed it. So I just wanted to give you this as a background so that if you get a slope that looks like a line as the tail goes out, then it's falling off like 1 over x to the alpha, where the slope is alpha, because it's falling off like x to the minus alpha. Minus alpha is minus the slope. So if alpha is large, then it's steeper. And if alpha is small, then it's more gentle. But so basically, to answer your question, if you take empirical question, you can say, does this look like a Levy walk? Or does this look like Brownian motion? So if you get data and you plot that data and then you fit curves, is this curve going to fit better or is this curve going to fit better? And you have a class of curves that look like this and a class of curves that look like this, where you might be fitting the slope to the data. Or it may be that a straight line doesn't fit and it's getting steeper and steeper. If it's getting steeper and steeper, you say it looks like a Gaussian process. If it's falling off like a straight line on this log scale, then you say it looks like a Levy process. Does that answer your question? And then you might say, why does it look like a levee process? Then you go back and you say, well, this animal has a foraging behavior where it's foraging within patches and then it's going to a patch. So you get lots of small steps within a patch, then you get a big step when it goes to a new patch. So you may say it's arising because of that. So it helps you understand the foraging behavior. Yeah?
The small tail, the positive slope at
Yeah.
the beginning,
Yeah.
the slow rate, is it because the alpha, the slope the alpha should be between 0 and 1?
Well, it turns down there because the animal may not make lots of very small. If it goes straight up, it means making more and more small steps, smaller and smaller steps. But the number of small steps may not be that high. In other words, the mean step size is not, or the mode step size is not going to 0. It's some other value, which means that the animal's basically moving around a lot, but it's doing so in small steps. And over here, the step sizes are very small, and every time you look at the animal, if it were a lot of the time in one place, then you would get a longer curve. But if it's keeping moving all the time, then you'll have that turning down. Does that? Yeah, okay. Anything else? Okay. So this paper, yeah?
Sorry, can you go back one slide? So you mentioned briefly before the frequency of the sampling frequency.
Yes.
Could this affect?
Yeah, absolutely. And this is what the next study is about. So this is an easier paper to read, and this is by Gauterstadt, J. Royal Society Interface 2012. So that's before that other paper I showed you. That other paper in Movement Ecology has got huge simulations and it's very detailed. This is kind of a first model, and then they got the idea of doing this in much more detail. But what you have here is you have data, and the data can be plotted at every time point, or it can be plotted at every 10th time point, or at every 100th time point, or every 1000th time point. So remember in the beginning I showed you how you can plot, you can look at your sample data, or you can plot you can look at every second point, or you can look at every 4th point. Well, here they're looking at every point, every 10th point, 100th. And if you do a log plot of frequency versus step length, you'll see that as you sample less frequently, so it's not unexpected, the average step size is bigger. So these curves are shifted. Now, here they're talking about beta, which is really, in the previous model, alpha. It's the slope of this, but actually I see that these are positive, so maybe it's the negative of alpha.
No,
huh?
Yeah.
Yeah. Here you can see that if you have, here's a levee walk. The thing about this levee walk is truncated. So because it's truncated, it actually starts to fall off here. In other words, its tails don't keep going on, because that means it has a maximum step size, which is very big. But you can see here that you get this levee process. And here, this looks like it's a levee process. But this data was generated by saying, I'm going to draw points not from a Gaussian distribution that gives me Brownian motion, but from 4 Gaussian distributions that are each shifted. So that sort of has the effect of smearing out a distribution, and when you plot the data, you might be fooled into saying, oh, the underlying process is levee. Here, the underlying process is not levee. The underlying process is 4 different distributions that you're drawing from at different phases. So you can say, okay, an animal has when it's kind of feeding within a patch locally, it has a very small distribution. Then maybe when it's more active, there's a second distribution. And then maybe when it's moving between patches, there's a third distribution. And then maybe when it's going from one zone to another, there's a fourth distribution. And it does this at different phases. And it may be different phases of the year, so that over the course of the day, maybe you have two Brownian motion processes, one where it's working within a patch and one where it's moving between patches. Then maybe over a season, a third process comes in where it ranges further so that it's going from a zone of patches over here to a zone of patches over here. And then maybe on a even larger time scale, there's some migration. Obviously, if there's some migration, you can't get lots of points coming out from the fourth process, because that doesn't happen very often. But you can see how, and we're going to talk tomorrow about what we call fundamental movement elements and canonical activity modes. And so each one of these Brownian processes probably corresponds to it being in one kind of mode. But when you do this, so the demonstration here is that when you look at data, be very careful what you conclude. And you have to go more deeply into the data. They even look at some different things where they say, here we have some random walk processes where you keep returning to a home. So you kind of range out and then you come back. You range out and you come back. And depending on the frequency at which that happens, you'll get different kinds of data. So if you want to read more about this, then you can read that paper. So we're not going to spend any more time on this, because as I said, the main thing that we want to do here is to get into building models. But before we get there, as a last thing that I want to do is I want to talk about things other than step size and turning angle distributions. Yeah?
Small question, please. What if part of the motion is Brownian and part can be Levy?
Yeah.
And is that going to confuse us, because the mix of the two,
Yeah.
you get the tail looks.
Yeah. So there are two ways to answer this question. Either somebody looked at it and it's in the literature, or you're going to have to build a model and say mix the two together and see what it looks like. So this is what they did, is they built models, they generated this data, they did a huge simulation, and they plotted this. And they said, what if we have 4 Brownian processes, each with different statistics, and mix them together? Now, obviously, the look of this depends on the relationship of the one Brownian to the other. You know, are they close? Are they far apart? If they're very far apart, then it would look very different as well. So this is where modeling comes in, to answer the kind of question you asked. And if you suspect that you have a Brownian processes added to a kind of a truncated Levy process, what would I expect the data to look like if the statistics of the Brownian relative to the statistics of the Levy are such that the means have this ratio or something? What would the data look like? Then I can't answer that question. I don't think anybody can answer that question without doing the simulation. So that's why we have these simulation tools, and that's exactly why we want to do this. So now there's the mean squared displacement over a population of individuals. So if you take an individual on a homogeneous landscape doing Brownian motion, then it starts out over here, Brownian motion. At any time, what's its distance from the origin? You do that for a collection of individuals, and so you've got a population of individuals, and you calculate the mean squared displacement that takes place over the day. And then you get, so this is the one that takes place over the year. So over a day, over 2 days, over 50 days, over 100 days, how far has it moved from day 1 or day 0? And so this curve doesn't look correct, because this one's the mean, and these are the 10th and 90th percentiles. Over here, the mean actually is below. I don't know what happened to this curve. It doesn't make sense to me. But anyway, you're supposed to get curves that look like this. And here you see that in Brownian motion, this curve actually should behave like 1 over the square, it should behave like the square root of time. So it's diffusion. Diffusion, the distance you move from the origin over time is proportional to the square root of time. That's a classic diffusion process. Brownian motion is akin to a diffusion process. If you've got a patchy landscape, you know, you might be within the land, all of a sudden you move, and then depending on what your structure looks like, you'll get different kinds of curves. This is real data from, again, Elk in Alberta, where you're calculating the mean squared displacement of individuals. So when you calculate certain statistics, then there are other things that you can calculate. And if you read the literature, there's certain things that are interesting, like in some of Run's papers with the vultures, there's certain things that we're interested in, and there's certain behaviors that they have in normal times, and then they also have these long-range forays, which would come out in some kind of analysis. So in the exercise that we do today, we're going to simulate different kinds of random walks. So Brownian motion is called a simple random walk, an SRW process. In a simple random walk, you have a step size distribution that's like Gaussian, and your angles of turning are uncorrelated from one step to the next. In a correlated random walk,
and they are uniformly distributed.
The
angles.
The angles, yeah. So they're uncorrelated and uniformly distributed on minus pi pi. In correlated random walks, there's some persistence in the direction from one step to the other. So you get your step size distribution, but you're more likely to go in the direction you headed, sort of persist directionally. You might make changes, but you're not going to just make a completely random change with regard to the unit circle. In a biased random walk, there's a particular direction on the plane that's kind of pulling you over. And so, for example, you might be foraging in a correlated random walk or an uncorrelated random walk, a simple random walk. But in the end of the day, you've got to get home. So you say, I might as well drift over towards getting closer and closer to my home as I do this thing. So there'll be a bias that pulls you in a particular direction. And in a biased correlated random walk, there's the bias, and the direction you're currently heading, though, you tend to persist in that direction for more than one step size. So what I've done is I've set up models, a model here that we can code up in Nova that will give me the next angle based on the previous angle. And if the next angle, based on the previous angle, you add to it a random component that goes from minus pi to pi, then it could be anywhere, then that would be uncorrelated. If this is less than, if A is less than pi, then you're heading in the same direction and you're perturbing it by something less than minus pi to pi, so then you're getting a bias coming in. And here, if this is your step size could be random, but if you have a particular direction so that when you're pointing in this direction, your step size is bigger than random, then it's kind of pulling you over in that biased direction. So here's a set of equations where you got a parameter A and a parameter B, and a parameter theta hat, which we'll set equal to 0, because we're just going to say the bias is in the 0 direction. We don't care where the bias is. But we're going to have a bias, so we've got two parameters, one that sets the correlation and one that sets the bias. And we're going to play around with these two. And then once we've chosen A and B, and then theta we're going to set equal to 0, then we can generate the next step size, the next location, based on the current location, the size of the step, and the angle that you're heading. And x takes cos of the angle and y takes sine of the angle. So that's just trigonometry. And so if you want to see an analysis of, well, we're going to do the analyses, and there's a reference here to McCulloch and Kane in Ecology talking about random walks and biased random walks, just if you want to read some background material. And we're going to choose parameters that firstly give us a simple random walk, a correlated random walk, a biased random walk. And we're going to repeat this simulation above many times. And we're going to look at the data and maybe plot histograms. And as I said, we're not going to plot the although we can use NOVA to plot histograms. We're not going to do it in NOVA because there are other things that you might want to look at as well. So you're going to take the data that you get and import it to MATLAB or whatever you use in order to build your distributions. So that's everything I got to say on my bit. I think we'll take a break and then Nick can talk about how to code the model up in NOVA. Does that sound like a good thing? Mm-hmm. So how long will we break for?
15 minutes.
15 minutes, okay. And will we come back here or go to the computer center? Yeah.
Can I annoy everyone and ask a question before the break? Or
yeah, of course. We love being annoyed.
Maybe it's out of scope for this talk.
Yeah.
I'm just wondering why in a simple random walk, I understand about the direction distribution, but why is the step size not uniformly distributed? Why is it Gaussian? If it's,
in the Brownian motion?
Yeah.
It depends on the process. So a uniform distribution so if you have to simulate, if you look at the average statistics and you draw from anything that is Gaussian or uniform, you see, multiple sampling of a uniform gives you a Gaussian in the end. You know,
Law of large numbers.
Not the law of large numbers, the central limit theorem.
Yeah.
So it makes no difference what you use. What's important is what kind of tails you have. So if you've got a compact distribution and you draw and you draw and you draw from that, after a while and you have a look how far you've gone, it'll look like diffusion no matter what you do. So it's much simpler. You can even have a fixed step size. And because the angle's arbitrary, if you've got a fixed step size, you know, you're jumping around like this and you have a look how far you've diffused, it'll look Gaussian after a while. It'll look like diffusion.
You're looking at the displacement.
Yes, if you're looking at displacement, yeah, it depends what you look at. But they all look very similar at a macro level when you look at how far is the individual displacement over time. It behaves like the square root of time in terms of that displacement. And so initially, that distribution looks different for each one, but after a while, it settles down into looking like diffusing out in terms of displacement. If you've got a variable step size, then obviously by the first step, you start at a point, and then after your first step, you can be an arbitrary distance. If you've got a fixed step size, you've got to be 1 unit away. Yeah. So displacement, yeah, but then after that, it begins to look like 1 over square root and look like diffusion. Yeah. Does that answer your question? Yeah.